So hello and welcome everyone to Webhub's Biotech Odyssey channel, and today we are going to discuss on the topic that is somatic cell nuclear transfer. So as the name suggests, somatic cell nuclear transfer. Here the nuclear transfer means the transfer of the nucleus, and the somatic cell means transfer of the nucleus of the somatic cell, right? So let us look at this statement, what it says. So, somatic cell nuclear transfer is a technique used in cloning which involves transfer of nucleus of the somatic cell into an enucleated egg cell. So, this technique is used in the cloning process, right, which involves transfer of the nucleus of the somatic cell into the enucleated egg cell. So basically, the enucleated egg cell means an empty egg cell in which the nucleus has been removed, right? So let us look onto the process that how this somatic cell nuclear transfer works. So in the first step, we are going to take the somatic cell of the donor, right? We will going to take the donor somatic cell that we want to clone, or the desired animal that we want to clone. So we will take the donor somatic cell, and then after this we will isolate the nucleus from that somatic cell, right? So after the isolation of the nucleus of somatic cell, in the next step we are going to take an egg cell from the female animal, right? After taking the egg cell, we are going to remove the nucleus of the egg cell, right? And after removing the nucleus of that egg cell, an empty egg cell, or you can say enucleated egg cell, is formed, right? So in the third step, you are going to transfer this nucleus of somatic cell into that empty egg cell, right, which will result in the formation of a fused cell. Now this fused cell is basically composed of an egg cell, and that earlier we had taken the egg cell containing the somatic cell nucleus. So basically this is called as now a fused cell, right? So now when the fused cell is formed, after this we are going to stimulate the cell to get divide into more number of cells, right? And then after this, after 6 to 8 days, or approximate, we can say that after 1 week, the cells will be completely developed into an embryo. The formation of the embryo will be occur. Now, this embryo will be implanted into the uterus of the surrogate mother, right? So the statement says like embryo will be implanted into the uterus of the surrogate mother. When the embryo will be formed after 6 to 8 days, then this embryo will be implanted into the uterus of that surrogate mother, right? Now, after this, the offspring will be formed, right? Now this embryo that we have inserted, this embryo will be now going to develop into a offspring. Now the offspring form is a clone of that particular animal, right? That will be a clone of that particular animal that we want to clone, right? So let us take an example. So one such example is your like dolly sheep. And dolly sheep is an example of a cloned animal that has been created through this technique, right? Okay, so now let us discuss about the key points of somatic cell nuclear transfer. That what are the important points of somatic cell nuclear transfer, right? So the first point is preparation of donor nucleus and egg cell. As told you earlier, that first of all we will take the somatic cell of the donor, and then egg cell from the female animal, and then we will isolate the somatic cell nucleus, and then after this we will create an empty egg cell, right? So this is the first step. Now in the second step, transfer of donor nucleus. The nucleus that we have isolated, that is the nucleus of the somatic cell, that will be transferred, or you can say inserted into the empty egg cell, right? So coming on to the third step is your fusion and activation. Fusion is like in the third step we have discussed that a fused cell is formed, and then it is stimulated to get divide and develop into an embryo. So this is your fusion and activation. And in the fourth step, the embryo will be formed, right? After 1 week or 6 to 8 days, the embryo will be developed, right? Now, in the fourth step, embryo transfer. In embryo transfer, we will transfer the embryo into the surrogate mother uterus. So, if you are telling about the somatic cell nuclear transfer, you have to discuss these 4 points, right? So next, coming on to the applications, that what are the applications or the uses of the somatic cell nuclear transfer. So the first is cloning of the animals. That it's simple, that we are going to create the clones of the specific or desired animals. The second one is your agriculture and livestock breeding. In agriculture or livestock breeding, for example, let's say we have a cow that has high milk production, right? So we can create the clone of that same cow to obtain that specific desirable trait, or we can say that any animal that has specifically resistant to a particular disease, or it is genetically resistant to a disease, and we want that offspring formed is also containing that similar properties of disease resistant, then we are going to apply this somatic cell nuclear transfer technique, right? So what will be happen, that if we create this, the reduction of the, or you can say the need of antibiotics will be reduced. So that will be a good point, right? So now the third application is your conservation of endangered species. So this is a very important point, like the animals which are going to extinct or will be extincted further, so you have to preserve it. So for the preservation of such species, we are going to use somatic cell nuclear transfer to conserve those species, right? So these are the applications of somatic cell nuclear transfer. Now next is advantages and disadvantages of somatic cell nuclear transfer. So firstly we will discuss about the advantages. So the advantages are your same, the reproductive cloning. You are going to create the clone of your desired animal and preserving. And the second advantage is your, you are going to preserve the endangered species. Another advantage is that by using this technique you are preserving the endangered species, right? And in livestock breeding you are obtaining your specific desirable traits, as told earlier, like a cow having a higher milk production, right? And you have to obtain that same in the offspring. So we are going to use that same technique. And for the disease resistant, a particular animal is resistance from a disease, and through this technique we can make the genetically resistant animals, and of that clone animals, we can say, so thereby reducing the need for the antibiotics, right? So these are the advantages. And now coming on to the disadvantages of somatic cell nuclear transfer. So the first disadvantage is like it has a low success rate, right? And the second is developmental abnormalities, like during the formation of the embryo, maybe some abnormalities can occur, which contributes to the disadvantages. Now the third one is your ethical issues. Ethical issues basically involve like manipulation of the embryos or destruction of the embryos, like that, or the abnormalities that are formed during the procedure of somatic cell nuclear transfer will be passed on to the offspring, which is not a good thing. So it also contributes into the disadvantages of the somatic cell nuclear transfer, right? So guys, this is all about somatic cell nuclear transfer technique. And if you found this video informative and explanative, so please like, share and subscribe the video. And thanks for watching.